dumping tons of graphs and images. Just wanted to give some brief introduction of certain neurodegenerative diseases and what am I doing at UC San Diego for last 15 years. And then couple of slides on the future technologies now we are developing to study brain diseases. So just a glimpse of what's the future of neuroscience research. So in this slide, you can see it's a nice cross-section of a brain. And brain, as we all know that it's a soft tissue, very well protected inside our skull, which is also covered with multiple layers of tissues and skin, right? And when you take out the brain and keep it on a dish, this looks like this. So this is one hemisphere of an adult human brain, right? So usually it weighs between 1.2 to 1.4 kilograms and largely made up of lipids and proteins. Now, if you take a slice of this brain and just look at under the microscope, this is of course a schematic. What we see is a tons of cells. All the cells in brain we call nerve cells or neurons, and numbers are tremendous. Almost like 100 billion neurons are present in one adult brain, right? And not just the number of neurons, but those neurons, what they do, they interact with each other. And that interaction is very, very essential for neuron to neuron communication. Basically, all our day-to-day life activities controlled by these neuron-neuron communications. And those communications, there are almost 100 trillion those interconnecting neuron points are there. And in neuroscience, we call them synapses. So in one adult brain, there are almost 100 trillion synapses are present. Now, just like every other organ, brain is also, susceptible to different diseases. Today, I'm not going to talk about infectious diseases that occurs to brain, but I'm going to talk about a little bit about, different regions of brains which are affected because of some, degenerative processes. But before going into that, I just wanted to give you just one slide brain anatomy where you can see in the schematics that different regions of brains are present, right? And each of those regions perform one specific task. Let's say, for example, speech is regulated by this frontal lobe, smell is regulated by another region, and then you have this temporal lobe that is essential for hearing and facial recognition. Vision is somewhere there, regulated by occipital condyle, the occipital lobe. So every region has specific tasks. But among others, there is one small, tiny region of brain is present inside this temporal lobe, which is called hippocampus. And hippocampus is the CPU of our brain. So basically that is the region where we is basically involved in learning. And that's the reason, hippocampus is the reason where memory is for day-to-day diseases. What are those diseases? Some of them are I'm listing here, like Alzheimer's disease, Parkinson's disease, Huntington's, Lewy body dementia, and several group of diseases are there. And as a whole, we call them dementia. So dementia is basically, dementia could be because of any of these different diseases, but at the end of the day, what happens when one person has dementia, the most important pathological phenotype is the memory loss. So most of the time, what happens, hippocampus is affected, right? So now, going to the next point, what I'm doing here for last almost more than a decade now, I'm working on Alzheimer's disease. And this is the most common cause for normal dementia. So before going deep into Alzheimer's disease, all of us, we are locked down, home quarantined, and many of us, we are watching good movies, right? So these are few movies. I have seen many of them, and I think many of you have watched at least one of them. If not, give a look. So Still Alice is an Oscar-winning movie. It's another movie, Hindi movie, came in mid-20s, Black. Amitabh Bachchan is there, and our very own Janu Borua from Assam. He made one telefilm, it's called Gusty Morning. So all of these movies, very interesting and themed to one reason. And all of those, that the main theme is of these all these movies is Alzheimer's disease. Basically, one or the another character in all these movies, they have Alzheimer's disease, and if you watch them, then you will realize what actually happened to those patients, and not just those patients, what happens to the caregiver, what happens to their family members. It's very well defined way to know about certain diseases. And if I talk about certain people who all are actually famous personalities who died due to Alzheimer's, we have our George Fernandez, who was the ex-defense minister of India, Ronald Reagan, US president. And at the end, because of this disease, he actually forgot that he was US president. And many other celebrities, they all died, due to this particular disease. Why is it important? If you see this graph, this will tell you why we should study these kind of diseases. If you see here, all diseases like breast cancer, prostate cancer, heart disease, stroke, HIV, all of them over the years, their occurrence or the percent increase is going down worldwide. But look at this graph here, almost 70% increase every year. What is the disease? It's Alzheimer's disease. So it's really, really dangerous. This is a worldwide data. So you must be thinking, what am I doing about this Western disease? Because, in India, probably it's not that frequent. That is what we initially thought about, but which is wrong. Actually, Alzheimer's is no longer a Western disease anymore. So why I'm saying this, because as you know, India is like highly populated, 130 crore total population. And out of 130 crores, we have nearly 10 crores of people, which is above 65 or older. Interestingly, aging is one of the major risk factors for this Alzheimer's disease. That means that after age of 65, the chances of getting Alzheimer's, it increases twice. And interestingly, after age of 85, that risk goes beyond that limit. So after 85 of age, 1 in 3 people actually will find having Alzheimer's. So according to 2015 data, nearly 50 lakhs people in India are having either Alzheimer's or related dementia. And that number is going to increase almost double by 2030. But problem here is in India, we still are concerned about infectious disease, tuberculosis, malaria, people are dying. So Alzheimer's is kind of a hidden problem. And as you see, this occurs to older people. Most of the time, doctors and even family members, we just ignore this as considering this is kind of a part of, inevitable aging process. So we ignore, but if you see the reality, it's much more dreaded than any other Western diseases. So I thought that, okay, this is not just, Western disease, we should definitely study this one. So what happens in an Alzheimer's brain? Okay, before that, going to that, I should also tell you that risk factor. Aging is one, and then environmental factors also trigger this particular disease. What are the environmental factors? Like stress, diabetes, food habit, high cholesterol. If we always have pizza and burger and all those things, it will induce somehow this particular disease. And then second thing is genetics. Even though 95% of Alzheimer's disease are sporadic, that means it can happen to anybody, but there are nearly 5% of people who get it from their parents or relatives, that is hereditary. And another cause is traumatic brain injury. Sometimes what happens, people get head injuries because of that fight or boxing or even bomb blast. So those people have been shown to, prone for developing this disease. So what are the symptoms? It's really important question, because symptoms of Alzheimer's are very common to regular, forgetfulness of what we always feel. So stages wise, it is not just, one stage. Doctors or clinicians have divided this Alzheimer's disease in 3 different stages: early, mid, and late. And if you go through different symptoms, I'm not going into all the details, but what happens when one person is about to get Alzheimer's, at least let's say around at the age of 65 or 70, they will start forgetting certain things. Like they will have trouble organizing or planning. They will forget what they have read this morning. They will start forgetting names. And eventually after 2 to 3 years of that stage, what happened, they will start, having delusions. They will need assistance for day-to-day life. They'll be having issues while learning new things. And then after another 2-3 years, that person will go to the late stage that is really severe. So what happens that stage is that they will basically, they won't know how to eat, how to speak. Even if you feed them, they don't know, they will forget how to swallow. So this is really complications. And most of the people die because of pneumonia and other diseases. So it's, even though it's slow, but it's going to happen. And it occurs once it is identified, it will take another just 4 to 5 years or 6 years at the max to, go to the last stage. So I'm just summarizing, this is the difference between, symptoms, what we see in day-to-day life to Alzheimer's. Now, what about the brain itself? What happens to Alzheimer's brain? If I take out 2 brains, one healthy brain you can see on the left and one Alzheimer's patient brain, you can see the difference, clear difference here. And in scientific terms, we call this empty spaces here, which is very visible, as brain atrophy, or in other words, degeneration of brain tissue. Basically, you have empty spaces all over the brain here. And if you take a cross section of this is what we see. This half is normal healthy brain, this half is having from advanced Alzheimer's. And you see here, basically hippocampus, which is essential for learning and memory, is completely kind of gone. It's wiped out. So basically neurons or cells are all dead by now. So this is what actually happens. And this is another section. What happens if you take a slice of Alzheimer's patient brain and look at under the microscope? What you see is this kind of deposition inside those brain area. And what these depositions are, they are called amyloid plaques. And these amyloid plaques are toxic to the neuron. Once they start depositing, they will start killing the neurons. And this is what a schematic would look like. This is a normal synapse, neuron to neuron, with connections with all these different synapses, millions and trillions of synapses. But what happens in an Alzheimer's patient brain, this amyloid deposition takes place just outside the neuron, and those amyloid plaques, they will basically destroy this connection and eventually it will kill the neurons. So what these amyloid plaques are made up of? These amyloid plaques are made up of one protein, very tiny protein called amyloid beta protein, or sometimes you can call that as amyloid peptide, or in short, A beta peptide. So what happens? Very interestingly, this peptide is basically generated from a parent protein called amyloid precursor protein. And all of us, including you have this particular amyloid precursor protein in your brain. But sometimes what happens, for some unknown reasons, 2 enzymes called beta secretase, or in short BASE1, and gamma secretase, they start cleaving this particular protein APP. This is the schematic I'm going to show. In normal brain, APP is present, just like every brain, actually. Every neuron has this protein. It's cleaved by 2 proteins. The scissors are enzymes, okay? Alpha secretase and gamma secretase, one after another, and then it forms kind of a couple of fragments. And this is non-toxic. In normal brain, your brain, my brain, this is happening. But what happens in Alzheimer's brain, instead of alpha secretase, another scissor comes, it's called beta secretase, it starts cleaving, and followed by gamma secretase cleavage. And this cleavage leaves A beta fragment inside the neuron, which is secreted out eventually. And those A beta fragments basically forms this amyloid plaque. This is the biology of generation of amyloid beta. So we know a lot of things within last like 20 years of research, 30 years of research. We know all these things, but problem is we still do not know why this happens. And you can ask one question, that what happens to this amyloid beta, why it forms aggregates? Because if you look at the protein structure, this is very, very hydrophobic. So it basically aggregates. Now question is, you can ask that, what about why there is no medication? Why there is no treatment? And you can basically say that, okay, Utpal, why don't you inhibit the base enzyme? And it's an enzyme substrate reaction. If you inhibit BASE1, you can stop basically production of A beta. But trust me, many companies, many researchers have been, trying to inhibit BASE1, and all of them failed so far. So we cannot inhibit BASE1 activity. Treatment failed. We cannot dissolve the existing amyloid plaques or beta. We cannot really do that. And we cannot really just open a brain and, start removing amyloid plaques. We cannot really do that. So there is currently no treatment is there. So we need to look beyond this enzyme inhibition theory. So in our laboratory, what we try to know that instead of inhibiting base, why don't we just basically stop APP and base interactions? Because they are enzyme and substrate, right? Plus two level chemistry, you can remember enzyme and substrate, they have to come closer. So we started thinking about if we can stop their interaction, then we can probably overcome this Alzheimer's issue. So to do that, we first generated two questions. First of all, we need to know where this APP and base are present inside the neuron, right? And then second thing, once we know that, then only we can look at what initiates these interactions, and eventually probably we can stop it, right? So first thing what we need is a neuron to study these diseases. So we use mouse pups. This is little gross picture, excuse me for that, but this is how we basically get neurons. We take P0, the mouse, the day get birth, we just decapitate their head and then take out the entire brain, and from that brain we basically isolate the hippocampi, and then we basically culture them in our dishes for our experiment. And this is one example, and this is the day 0 when you take out the neurons from mouse brain. They look like that, and eventually after 14 days, 15 days, they grow their processes. And this is what I have stained here. You can see nice neurons connecting with one another. And one slide, this is a single neuron I am showing you, where you can see nice cell body. It's a textbook kind of picture I have taken you for your, it's easy to understand. It has a cell body, you have multiple dendrites here, you have nice long axon here. So this is what a neuron look like when you culture them outside the brain on a dish, right? So once we have this system up and running in our lab, then we thought that, okay, let's look at the APP and base where they live inside the neuron. So to do that, what we did, we used normal, regular recombinant DNA technology. You all guys know all these technologies. Your teachers have taught about it. So basically, we have taken out the APP gene, which we have fused to a green fluorescent protein to give it a green color. We have taken out base gene, we fused to a red fluorescent protein that gives it red color. And basically, you just throw them inside the neuron, and eventually you will have APP as a green dot, base as a red dot. So it's basically, you can see them. And this is a movie I'm showing you. This is a small portion of a dendrite here. And I have expressed APP tagged to a green fluorescent protein. And I just looked at under the microscope. And this is what you see. So see this white dots, they are actually green. They are APP protein moving inside the live neuron. So see how nicely they are moving from one direction to another. So this kind of studies, we do live imaging of different proteins in all those neurons, either axons or dendrites. So once we have these tools and techniques and neurons established, then we went back to our main question that where do APP and base live inside the neuron? And we express this red and green two proteins. And you can see here, when we overlap that, we found really interesting observation here. What we have observed that these green dots representing APP, they are segregated from red dots, right? They are segregated from the red dots. That means APP and base at a resting state in a neuron are segregated. If our observation is correct, that also tells indirectly that why not all of us get Alzheimer's. Probably nature has developed a way to keep them separated. Otherwise, all of us would have got Alzheimer's, right? So they are together, enzyme and substrate, they have to come together. But at normal stage, they are segregated, they are separated. But there are people, some people do get Alzheimer's. Why so? What happens? Then we started asking these questions, and we did a lot of literature research and found a couple of papers where people described that certain brain activity in certain areas, those activity-induced areas are enriched with this kind of toxic deposition, and those areas as a whole is called default mode network. So default mode network, where those areas particular areas there, those are basically enriched with amyloid deposition. So what is this default mode network? These are certain regions of brain which are activated when you are basically not doing any specific work. Let's say you are lying on a bed, you are closing your eyes, but you are not thinking something constructive, no specific thinking. You are just randomly in your thinking. Let's say you are thinking about money and all those things, which is kind of one way you can say daydreaming, or sometimes stress. So this kind of cues, environmental cues, activate this default mode network, which is kind of prone for amyloid deposition. So we thought that, okay, can we basically recapitulate this default mode network activation in our cultured neuron and then see what happens to APP and base? So to do that, we went back to our original experiment where we saw that APP and base are segregated. And then what we did, we generated neuronal activity, extreme neuronal activity, and looked at look at under the microscope. And what we have observed here, green dots and red dots are now coming together, giving you yellow dots. That means APP and the base, enzyme and the substrate, they are together. Not just that, it also generating a lot of more toxic amyloid beta. So this is kind of a fundamental observation which we have observed that at resting state, normal brain state, normal human, APP and base are segregated, but when you induce neuronal activity, there is an increase in APP-based convergence and more amyloid beta generation. So how is it possible when two things are separated? We also studied that, and we found out that endocytosis, all of you, I think, I'm sure you know what is endocytosis. What happens when you induce neuronal activity? APP undergoes endocytosis, and then it converts into a base-containing endosomes. And we have used different inhibitors to stop this process. So we published this paper, you can go through it, or you can ask me what exactly in more details. But these are fundamental observations, and we're very happy we celebrated these discoveries. But just like any other science, this generated more questions. The questions we generated then if everything is known, all the mechanism, why can't we stop Alzheimer's? Why there is no available treatment? The one main reason is that the model system so far we are using, mouse brain, mouse neuron. And we are not mouse, right? Humans are not big mouse. So end of the day, we need to repeat basically all this experiment and basically extend all our experiment in human neuron. But the problem is, can we get human neurons? So we cannot just open in someone's brain and get some tissue and then culture them to get neurons, because all other organs are available. We can open, we can do surgery and get liver tissue, culture liver cells. But in brain, you are not allowed to do. People die. So it's kind of not permitted. So option only left, can we make human neuron? Can we really make human neuron? So actually nowadays we can. Where from? We can actually nowadays make human neuron from your skin cells, all of yours. If you give me a skin cells, little bit of skin tissue, I can make neurons out of it. So how do we do that? You just take just little biopsy tissue, skin, and then add certain protein factors, it will become pluripotent stem cells. Pluripotent means those cells, it can form to any lineage. It can, if you want, you can make them liver cells, you can make kidney cells, and not just that, you can make neuronal stem cells. So neuronal stem cells is the precursor, and if you had certain factors to neuronal stem cells, they will become adult functional human neuron. So this entire process, it takes nearly 2 months of time. Within 60 days, if you give me some skin cells, I can make neuron out of it. It looks very simple, and Shiny, Yamanaka, and Gordon, they got Nobel Prize in 2012 for discovering this particular pathway. So what I did, I wanted to use human neurons and study what I was studying. And luckily, in our university, Larry Goldstein is here, who is a pioneer of stem cells and neuron development. I collaborated with him. And what I did, I took skin cells from normal healthy patient and skin cells from Alzheimer's patient. I made this kind of neurons. This is a human neuron. And right now, I am studying APP endocytosis, that mechanism I showed before. And I have already obtained really interesting results. Certain key regulators of endocytosis are missing in Alzheimer's patients. Probably this is going to give us some clues about this particular pathway. So far we are here. Probably next time when we talk, I'll be able to tell you more about this particular process. But 2 more slides. Using this technology, where it is basically taking us in the future. Can we make a brain, full human brain in a dish? Can we really do this? Actually, we can probably. So it's called brain organoids nowadays. You can see this nice ball of tissue. These are lab made, and this is in a dish what we are doing here. We call them mini brains. So basically the same technology, we get skin cells, we make iPSC or stem cells, and from those stem cells, we make these mini brains. So what the use of these mini brains, we can basically, they are better model for disease. We can use them for, drug discovery. And not only that, probably in future, you can take out, you can basically transplant these mini brains to Alzheimer's patient, right? So this is what already people are trying to do in stroke and multiple sclerosis, ALS. People are doing all these things. So I think I'll stop it here. I think I have taken my 30 minutes, and at the end, I would like to thank you all once again for your Sunday time. And I would like to thank my team here. You can see, and you can contact me if you have any specific questions. And this is my email ID. Feel free to talk to me. Not a problem. Okay.
Thank you, Utpal, for this wonderful presentation. In fact, it was very interesting, the concept of mini brain. I hope my student enjoyed. So we will look forward for more interactions in near future. So I hope you can make some time for my students.
Definitely.
So,
thank you, Utpal. Thank you very much for your time.
Yeah. Great meeting you, and good luck to all of you students.
Thank you. So all the students, you can see sir's email here. If you have any question, you can directly email him, or you can mail, you can send the questions to me or madam, so we can also forward him the questions. Thank you everyone for attending
welcome.
this. Yes, thank you everyone for attending this half an hour webinar. A very warm goodbye to everyone. Thank you.
Thank you. Take care. Bye-bye.
Thank you, sir.